Again, it's very much subjective which population database you should use. Previously, people were using 1,000 genomes, and there was also something called exact uh, consortium. So exact consortium is this one. So now the data has been again migrated to Nomad. So but what happened in exact consortium is that they had some 60,000 individuals, healthy individuals with exon sequencing data. So from there we can calculate the population frequencies. And imagine you found a variant, and this variant is also very common in healthy population. It means that it's not really going to cause the particular disease. So that is the whole idea here. So if you have a variant and the variant is pretty common in the population, then you can exclude that variant as a pathogenic variant. And this is one of the strongest criteria for excluding a variant. For instance, imagine you have a variant and the variant is present in more than 5% of the population. You exclude that variant as pathogenic. Rather, you classify, you can strongly, it points out to be a benign variant with that one criteria alone. So 5% is the usual cutoff I mean, so if it is more than 5%, it strongly points out to a, very strongly points out to a benign variant. So which means that by default, your pathogenic variant should be rare in the population. Now, when I say rare, there is no one single cutoff allele frequency. It is heavily dependent on the individual population. That is why population-based genomic studies are important. So that is why everyone is eagerly looking for the variant data from the Indian Genome India project, because as of now we have some information about some variant frequencies. This is based on some small-scale sequencing that is done by IGIB or CDFD, etc. But having said that, we don't have a healthy population level large-scale frequency data. So this is very important because this one single criteria can help in filtering a large number of variants. So I can show you here, for example, can you see all these variants? So the first one out here, it is present in 95% of the population, which means that, you know, rather if you annotate it now, that will be like the reference allele only. I mean, it's present in most of the population. Something that is present. So this is all in percentages. Okay, so 0.04. I mean, so it's been 4%. Something that is present in 66% of the population. So all of them will be excluded. So if you want to apply a filter here and you want to do that. Sorry.
Take a allele frequency of how much?
So it's up to you. But what I
5%.
Yeah, so we will keep everything that is less than 5%. I mean, we can do it in Excel, but only problem with Excel and Mac is they are not really compatible. So if it had it been a Windows system, I can filter and show it to you also. Like what the number will actually decrease to.
I saw some numbers as 0.0003, 0.0003. So that would be something which I would need to see, but something as a 0.22 I may not want to see.
Yeah. So you can see here. So this is like 1.39 into 10 raised to minus 5. So very rare.
That's a very rare.
Some things are dots. Dots means they're not there in that particular database in Nomad at all. So Nomad, if you remember, they have sequenced a few lakhs of exomes. I think 3 lakh or something, and 6 lakh or something, and then they have close to some 70,000 genomes. So this is, and you can see here, they also, Nomad also has individual population frequency. So we can see this is in Africans, and this one is in Americans, and then some of them will be in Ashkenazi Jews, and I think Finlanders, and I mean, I don't know. So they do have some data from different populations, but there is nothing specifically for South Asians here. So South Asians are underrepresented Africans were underrepresented, but nowadays there are a lot of African genome projects for the simple reason that because the humans have originated in Africa, they think that a comprehensive African genome project can point out to a lot of diseases, and and we can understand the biochemical base of a lot of disorders from there. So that is there. And similarly, you also have what is known as nomad genome allele frequency. So we can use this one also, and we can filter. And similarly, we have other sites also, other sequencing projects also. For example, there is this company which is known as Regeneron. So I mean, Regeneron is one of the largest sequencing companies. Actually, they are a pharmaceutical company. So what they do is they do sequence a lot of genomes and healthy populations, etc. And some diseases also, very large scale. And then they try to derive information for drug development for this one. Similarly, another major study that these days you have is what is called UK Biobank. So that is also there, UK Biobank. So UK Biobank, they are sequencing again lives of individuals in UK. I think that is also more than 6 lakhs, I think, or even more. And there's also something called All of Us project. This is by the NIH. So they are also trying to get more than 1 million genome sequences done. And this will all come into play later when we actually apply this one. But having said that, it's very important to have our own population databases, because we cannot rely on external databases, because the alleles that we get are very much, the variation that we have in our population will be a lot different from variation that you find in some other population. So this context is important right out there. But right now, since we don't have anything, we'll use these ones. So that is population databases. The other thing that we can do, but not in an annotation file, but what we can do is we can also look at Mendelian segregation. This is a very important criteria for variant filtering. What do you mean by Mendelian segregation? There are a few criteria. For example, this is why the pedigree is also important, and this is why when we say a trio sequencing or an extended family sequencing in some cases, or the sequencing of a non-affected sibling, for instance. So a lot of these things can be important because this will really help in prioritizing the variant. For example, the child is having features of an autosomal dominant disorder, autosomal dominant. Both the parents are unaffected in that scenario. So both the parents are unaffected, but the child is having phenotypes of an autosomal dominant. Yeah, it is because of the spontaneous or a de novo mutation. So in that case, what you can do, you can look for variants that are present in the child but absent in the parents, right? So only those variants will be important. So any common variant that is present in the parents and the child can be removed from your analysis. So you can imagine, so a large number of variants will get filtered in that particular process.
I didn't understand that. You're looking at a child with a problem. You want to see the mutations which are there in the SNVs which are there in the parents and the child together.
Yeah.
Why would you want to exclude them?
Because the parents are not, you're thinking of an autosomal dominant disorder.
You're thinking of autosomal dominant disorder and the parents don't have any symptoms. So if there are common SNVs, you will ignore them because it's not the disease causing one.
Exactly.
Got that.
And same, in a similar manner, you can have your own approach for X-linked disorders, and also, obviously there will be looking at X chromosome, and also you can have an approach for autosomal recessive disorders. So there, you'll be more interested in heterozygous variants in the parents and homozygous variants in the offspring. like that,
A
right?
very
So
tangential question, not related to this. Have you registered in the PCPTND Act, or you're beyond that because you're not looking at this as an unborn children?
we are not reporting anything, sir. So
Okay.
uh, online. We are not doing any patient reporting as such, so we don't. I mean, for research, we don't need that.
Okay.
Right. Yeah. So that is Mendelian segregation. So Mendelian segregation, remember, with one VCF file alone, you cannot do it. For that, obviously. So that is yesterday when I was telling you about joint genotyping, that comes important because you can look at all the variants, right, in the same VCF file and same annotated. So here you can see in this annotation file, at the one of the last columns out here are the sample details. What is happening in individual sample? So each sample, what exactly is the variant? So that is already here. So imagine if it is a trio sequencing, you will have 3 samples here instead of so many. This is actually from a slightly large scale study, so that's why it's 30 samples. But if you're doing it for a patient, obviously it will be 1 sample or 2 samples or 3 samples, depending on how exactly you're approaching. Okay, so that is Mendelian segregation. Now these 3 things. Okay, so that is how you filter. The other one is also important. For example, if the variant is already reported in a non-clinical database, and when we say clinical database, actually this part is a little tricky because what we are talking about is ClinVar. And so ClinVar, for example, if you got a variant and you want to know whether that variant is really pathogenic, you can search for that variant. For example, I'm checking, CFTR. So it shows a list of variants that are pathogenic, likely pathogenic, uncertain significance, and so on and so forth. So even you can filter out the variants that are pathogenic right here. Now, only problem with ClinVar is that ClinVar is not a curated.
Boss, can you just go 2 steps back? I just missed this. How did you reach this, and how did you filter on pathogenic and all that? Can you just go 2 steps back?
I'll come to this one. Right now, how do you get to pathogenic? What I'm telling is, suppose if you have a variant, and the variant, I'm talking about this attribute.
No.
So we already done genomic context, population, and Mendelian segregation. I'm talking about another criteria for filtering or when it comes to prioritization. If that variant is already present in a clinical database as a pathogenic variant, your variant is the variant that you identified. Someone has already reported it as a pathogenic variant, and it's present in a well-known database. Then obviously your variant is pathogenic. That is a simple extrapolation. But there is a catch there. The catch being ClinVar. So let's say I am looking at ClinVar and I have a gene and I am looking at single nucleotide polymorphisms here. Now this is CFTR. So it's telling me that this variant is pathogenic. For example, in the coding region, the initiator within, right, the first one, A to G, is a pathogenic variant. What do you mean by that? That means if I am finding the same variant in my patient, then I'm pretty sure that it's a pathogenic variant for the simple reason that a well-known database has reported it as pathogenic. The catch here is that ClinVar is not a curated database. People can submit it. It does not undergo any quality filtering or a confirmation as such by anyone. So it's just a database where there is no regulation as such. So we presume that people who are submitting it are genuine and they are actually reporting the true variants or something like that. So nowadays what they have done is you get the stars also out here. So anything that is having a large number of stars, it means that it has been reviewed by. And can you see? Sorry. If I'm hovering my mouse out there,
It shows something.
it's showing.
Yeah, it shows something.
Yeah, it's showing something. Yeah, it says that reviewed by an expert panel. So that's what you mean by that. So review status. So number of stars 4 means it's a practice guideline. 3 stars means it's reviewed by expert panel and so on. So this is how that star symbol is there. So, but remember that for a lot of variants, if it is a 1 star or a 0 star, then you should be a little skeptical because it's not at all well curated or verified. But this is how we check for it. So if it is a variant that is known to be associated, for example, I can give you a simple example. I'm doing a CFTR and I get a delta 508. That is very well known to be associated with the disease, and so that is undoubtedly this one, right? So even we can check here 508. Sorry, 508 will be in a different page altogether. Display options. Where is it? Probably in the next page. Because this is in the order. So here we have gone.
There are so many mutations in the variants in the CFTR region that is pathogenic.
At least those are reported as pathogenic, yes.
By
Which is possible because it's a very well known gene for with a strong phenotype, right? And one more reason, since you have asked that question, I'll tell you the other reason why you see a lot of variants out here. Because CFTR is something that affects rich people. Cystic fibrosis is something that affects rich people, right? So that is why you have so many drugs for CFTR, cystic fibrosis. So you have that modulators, effectors, and all those things, right? Potentiators and all those things, because a lot of money was pumped into CFTR research because it affects Ashkenazi Jews and who are rich people compared to sickle cell, which affects poor people. So that is how it goes. So there's a discrimination that way as well.
In India, is CFTR a common condition?
CFTR is there. It's not uncommon. I think people do get it. They still work on that immunoreactive trypsinogen and all those things as screening for. I think it's there. 508 is where is that one? 507 termination deletion. Oh, sorry. You will not find it here because I have already. I didn't mark deletions, right? If I do deletions here, you will find 508. Yeah. So 508. No, not 508 deletion. No, that's a coding region, not that one. You're talking about this one, yeah, right. For example, this is a well-known variant, right? 508 deletion. This one. So here it says that it is pathogenic and 4 stars, which means it's a practice guideline. So it's for sure that this is pathogenic. So that is about clinical databases. And the next thing, the next 2 things are. One of them, you still get the information from ANOVA. So that is known as informatic predictions. What do you mean by that? Imagine you have a variant, and that variant. So there are a few things that you can look into. For example, you can look into the nature of the variant. So let's say an isoleucine is getting substituted by valine. Both are hydrophobic amino acids, both are branch chain amino acids, almost similar in size. You might not really think that it's going to do anything to the protein as such. On the other hand, imagine just like sickle cell, where glutamate to valine, polar to non-polar, or a histidine residue which is present in the catalytic site, which is involved in catalysis, getting replaced by a residue of a different charge or no charge at all, or a cysteine residue which is involved in a disulfide linkage getting substituted to tyrosine, which cannot form a disulfide linkage, so that the protein structure is getting affected, or tryptophan getting substituted. So tryptophan is a bulky amino acid, and now it's getting substituted to something else. Or take collagen, for example. You have glycine, which can be accommodated within the triple helix, getting substituted to any other amino acid which cannot be, which will disrupt the helical pattern. So we can make some predictions based on the nature of the substitution. For instance, right? How exactly it is going to affect the protein. So there are tools to do this predictions, informatic predictions. So few common ones that used to be prominently used was one is called SIFT. So SIFT stands for sorting intolerant from tolerant. What do you mean by that? So you have some variants that are tolerable, some variants that are deleterious. So this tool will predict based on a lot of this information between a deleterious variant and a tolerable variant. So how exactly you do that? Like I told you, so you can have a few different criteria for this one. I'll give you another example. So suppose if you have a variant, the variant is present in a very well defined domain of a protein, and the domain is conserved throughout evolution, which means that the domain, that particular amino acid structure sequence is important because when you cannot have variations in that particular sequence. So if you take humans and monkeys and mouse or some other organisms, in all of them, if that variant is conserved, then you know that that's a very crucial, critical residue, amino acid residue. So you cannot really have a variation in that without deleterious effects. So this is usually done by these prediction tools. Now, the problem with prediction tools is that, as the name suggests, these are prediction tools. They are based on some protein models, some sequence homology information, etc., and by looking at the domains, etc. So you're not really sure. I mean, these are not functionally validated in the actual real life scenario, in actual experimental setting, but still they help in predicting whether a particular variant is damaging or not. And we use this information also for prediction using these informative computational prediction predicting models for potential deleterious effects. Nowadays, what we commonly use, so this SIFT was used until recently, but nowadays, after Google came up with AlphaFold, for which the Nobel Prize was also given. So you have a similar tool based on Alpha tool, which is known as Alpha Missense. So Alpha Missense has a pretty huge catalog of proteins, and along with that, there is also another tool which is known as Revel. So Revel is a tool. Alpha Missense is another. These are all different predictive tools to do this particular process. So we can use this one and we can do this, but there is a catch here also. Like I told you, I mean, even if we are using Alpha Missense or any of those ones, still they are predictive models. I mean, even if we are using the latest AI models to generate this, still they are AI models, so they have their own limitations. So we will not trust them blindly, but we still use them for filtering the variant. So that information is also here when it comes to the annotation. So we have Alpha Missense here. So it has it will come with a score, and some sort of anomalous score. Here you can see. So I think they have. I think it is benign pathogenic, and A stands for most likely ambiguous, I think, and a dot means there is no information. We can exclude all the benign ones here, for instance. And
Dr. Ashik, I think
yes, sir.
Singhal is trying to join in.
Oh, all right. Yeah. Right. So that is basically alpha.
Dr. Singhal, welcome. You can mute your audio.
Thank you, sir. Thank you.
Single is walking from somewhere to somewhere. Okay, is in the car. Okay.
Car.
I hope you're not driving.
He is driving and listening.
Oh, seriously? Don't.
I am on the other side.
Okay. So that is basically alpha. I mean, so that is one prediction. So we generally use more than one tool for this, and we exclude the benign variants in this particular process. There are so many tools to do this. Okay, so there is something called
So the annotated thing has gone to all these places and taken up all the information, given it to you. So you don't really need to go and try each one of them over there. It has done it for you.
Yeah. So that is exactly what annotation does. So you can add this information automatically to your VCF file during annotation.
So all these steps that you said, genomic content, population dynamics, Mendelian segregation, known clinical database, informatic prediction, and functional annotation, all these informations are already come into this Excel, this filter pipe
No.
you're doing.
Few things will not be there. For example, Mendelian segregation will not be here because that is completely dependent on your clinical context.
Okay.
So that will not be there. I mean, you do get the information, but it will not. I mean, it will be there, but it's up to you to, because all I am seeing here is this whether it is homozygous, heterozygous. That information I'll be having it here, but it's up to us to filter it and interpret. But it will be here in this file.
Okay.
From our samples, right? And similarly,
information is here. You need to apply your filters. It's not applying the filters.
yeah,
And giving you
clinical databases are also here. I mean, for example, I mean, we can put the information from ClinVar, etc. But you can do that. I mean, you can add something. I think we do have some data. So this is a Regeneron allele frequency. I didn't use it for filtering, but you can use this one also. This is another tool for filtering. And then something here. Yeah, some of them. So you can see this particular variant is associated with congenital myasthenic syndrome. So that information can be there, at least for a few, not for all the variants, but at least for a few. So this one, Charcot-Marie tooth disease, and some information you can still get it here based on clinical relevance. So I think that is all about informatic predictions. And the last one, which is actually not there in the database, but probably one of the most important one is a functional annotation. And this is where a lot of research goes into, because what do you mean by functional annotation? You have identified a variant. You really want to know what exactly is happening to that particular protein and to that particular protein in the presence of the variant. And you want to know whether your variant is actually damaging, or for example, in case of an enzyme, it is going to affect the activity. In case of a receptor, it is affecting the signaling, let's say, or in case of a structural protein, it's going to affect the structure of the protein or something like that. And